Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Vinod Nair. Listeners, Vinod is Associate Laboratory Director of the WADA-Accredited Sports Medicine and Research Testing Laboratory, also known as SMRTL. And today we are looking forward to discussing his career, some progress that's being made in automation in anti-doping laboratories, and some of his recent research. So, Vinod, we are thrilled to have you with us today. How are you?
I'm doing well. Thanks for having me.
Well, thank you for joining us. And to give our listeners maybe a little bit more detail in terms of your background, I'd love to talk first about how you initially got interested in anti-doping science and perhaps, Vinod, how you found your way to this current position there at SMRTL.
I'd like to be able to give you some poignant, pivotal moment, but like everything else, it was just a bit of luck and coincidence. So the Salt Lake Olympics happened in 2002, and I was a graduate student at the University of Utah at the time. And for some reason, I wasn't relocated during the games. So I somehow happened to be right in the middle of the Olympic athlete village. So that introduced a bit of unintended familiarity with the field. And then a few years later, when it became time for me to graduate and get a job, I realized that SMRTL was established and it was just down the street from my old lab. And around that time, Floyd Landis also tested positive for synthetic testosterone at the Tour de France, which made quite a bit of news locally as well as internationally. Locally, because Salt Lake has a very active biking community. SMRTL didn't do that work, of course, but the whole topic was buzzy and topical enough that I thought it might be fun to go work there for a little bit. I had a good friend that was working there part-time, and he asked them if they were interested. So I lined up an interview, and they offered me a position. It was meant to be a short-term thing, but it's been 17 years now, so I think this one might stick.
I would say so. So now, before you started working there at SMRTL, had you had an interest in sports or were you an athlete yourself?
I was not. I was interested in sports as a casual watcher, but I wasn't an athlete myself. I did keep abreast of recent happenings, and the Olympics and things like that are always interesting to the casual viewer, but I had no active involvement in sport. I was familiar with some of the analytical techniques they used for anti-doping, and I guess that was my only real familiarity with the field.
Well, it sounds like the field has pulled you in and captivated you. You mentioned you've been there now at SMRTL for 17 years. So maybe could you share with us, what do you do in your current role there at SMRTL, and what is the scope of the work that you do every day?
So as you mentioned before, my current position is Associate Laboratory Director, but I've had that since 2009 or 10. So it's either the Peter Principle in action or a remarkable lack of ambition. Either way, I'm still involved in a lot of the day-to-day aspects of anti-doping, especially with data review as it pertains to reporting positive findings, which always have a lot of scrutiny associated with them, or doing method development sorts of things. But my job does allow me the flexibility, really, to float about the lab from time to time and work on ad hoc projects, such as creating efficiencies or experimenting with automation. Some of these things have low success rates, but that is just the nature of the job. And I guess I must be doing enough that they've kept me around.
Oh, certainly.
Occasionally, we do have a spate of unusual findings related to a certain compound. And that might involve the need to do some controlled administration or research studies to explain those findings and to contribute to the literature around those substances. I'm involved with some of those as well as time permits. Once in a while, I also need to testify in arbitration proceedings defending the lab's work or write expert opinions for our clients.
Very interesting. And I think the everyday work in the lab is maybe similar to what a lot of scientists out there might be familiar with. What have your experiences been like kind of on the legal side doing the testifying in these cases?
It's been generally good. We usually have a little bit of prep ahead of time, so we know at least what questions are going to be asked of me from the prosecution side. What you might get from the athlete's defense side is a mixed bag. It could be anything challenging the quality of the work or our motivation to produce such a finding. Usually these can be refuted with a few well-chosen statements, but I have had the odd allegation made about the lab or about my career itself. They've usually gone away and the panel hasn't considered them very seriously, so I'm okay with that.
Yeah, I think that would be stressful to sort of be on the stand in the courts, but you sound like you handle it well, calm, cool, and collected. And I know in anti-doping, there can be some pressures, particularly to have this fast, cost-effective, whether it's sample preparation, processing, analyses, all of these things are very important. So Vinod, can we go into some detail on just why this speed is critical for anti-doping labs and just what are some of the more time-intensive steps that you have to do when it comes to the routine side of anti-doping work?
Rapid turnaround is important because it enables efficient results management. At spotlight events such as the Olympics, or even in playoff type situations in professional sport, rapid turnaround facilitates a clean athlete lineup for the subsequent event. But even in out of competition or routine testing, fast turnaround can help management of adverse or suspicious findings. Depending upon the drug in question, a quick report can enable the testing agency to collect a follow-up sample, which would then add an additional data point to support the previous finding. An example in case is when SMRTL had our first growth hormone positive. Now, these are rare to begin with, and this was quite a while ago, so they were even rarer. And that's always a nerve-wracking thing for a lab to report the first finding of a particular substance, just because of the high stakes involved in that process. However, because we turned that first test around so quickly, the testing agency was able to go get a follow-up sample from the same athlete, which also produced a positive. And this greatly increased the confidence in the original finding itself. So rapid follow-up is one of the outcomes of rapid turnaround, and that can be very helpful in a lot of these cases. With respect to time-intensive steps in the laboratory, most of the routine, everyday stuff, like the screening that all our samples get, has been optimized in most labs to be high throughput and fast turnaround. But specialty tests, like the test for erythropoietin or the test for synthetic testosterone, the IRMS test, they're very involved and it requires a lot of sample preparation and workup. And when you couple that with an 8-hour workday, the analysis can stretch to 2 or 3 days. And that's just the nature of the test itself and wanting to go home at a reasonable time.
Right.
On the other hand, you could have some very expensive kit-based assays where you have one shot once you commit the kit to a test, and a lab might want to wait till it has enough samples to fully utilize that kit to keep the costs manageable. So that can also delay samples sometimes.
Definitely. And I think in this response that you just gave, you highlighted that cost is an important factor as well. So you mentioned optimizing use of these kit-based assays as something that can help control costs. Are there other things that anti-doping labs can do to, again, try to keep those costs under control?
We have to realize that cost is just one of the facets of the testing. There's also speed and quality of the testing. And the old adage is that you can only have 2 out of these 3. So what we're trying to do is achieve all 3 here. But there's no silver bullet or turnkey solution to minimizing cost. It's often a series of small incremental steps that cumulatively, with either time or with economies of scale, can lead to cost savings. So an example here is optimizing your batch sizes so that the instruments have as little downtime as possible. Or a lab might switch from old school cartridges and tubes and vials to 96-well plates. So the miniaturization is helpful. You have less consumable cost. You generate less plastic waste. You have less technician time. And miniaturization like that also opens the door to automation, which, while it has an initial capital investment, leads to long-term gains in cost. To stretch that evolutionary metaphor further, sometimes you can have technological changes that lead to a giant step immediately. So new technology coupled with instrument sensitivity can let you combine multiple assays into one, which is an immediate cost and time savings impact. But it's not always steps forward. Sometimes you also have steps back because the prohibited list keeps growing all the time. And all it takes is for 1 or 2 new substances to be added to the list, which don't fit neatly with any of your carefully optimized workflows. And then you have to introduce another assay, which negates some of the things I mentioned earlier. So it's a balance between comprehensiveness and cost.
That makes sense. And I think the decision of when the value of automation will outweigh the cost can be a difficult one. So can you give us some insight into how does SMRTL incorporate automation in the work that you do there today?
Our approach to automation is not so much as a goal in and of itself, but rather a way to make our team's life a little easier. So our belief, my belief anyway, is that an experienced and well-trained team is our most precious resource. So the focus is not on replacing them, but rather freeing them up so that their skills and their experience can be utilized better. So at SMRTL, automation is also done with a little bit of a pragmatic outlook. And sometimes sort of a hands-free walk-away solution can be really expensive, but eliminating the most tedious tasks with low-tech automation can get you most of the way there. I'll give you an example. So our everyday processes that are uniform from beginning to end are the most amenable to walk-away automation. We can invest the resources and it takes a little while to set up, but after that, it's very, very routine and uniform. So those are the types of processes that work really well with hands-free walk-away automation. But if you consider a simple step such as receiving samples, that's still a manual process at SMRTL because we receive samples from many different clients who produce different types of bottles and shapes and caps and covers. And it's really hard to find a single solution that can deal with all those different types of bottles. It can be really expensive as well. So that's still a manual process, but by configuring our LIMS, our lab information management system, and by using large volume programmable pipettes, we've made it so that a single barcode scan will both register the sample and generate all the number of aliquot labels required for the testing. And by using the programmable pipettes, all of the aliquots can be generated in a single aspirate and dispense action. So it's still manual, but we do use some low-tech automation to speed processes along.
Oh, that makes sense. And it sounded like the examples that you gave and kind of the description that you gave really applies perhaps best to routine work. Do you use automation at all in the research side of the work that SMRTL does?
Not as much. The thing with automation is that it requires a little bit of work to set up and validate, obviously. And the places where it's most useful are with routine, everyday things that you'll keep doing day in and day out. With research, the number of samples involved can often be smaller. And it's sort of a niche operation where you have to develop a new assay. You do have to validate it, of course, but then it's often not worth the effort to set up a whole automation process when you might only have 496 well plates worth of samples to analyze. So a lot of the research that we do is often on a small scale just because of our resources and because we have everyday production work to accomplish. So we don't tend to use a lot of automation in research, at least not yet.
That makes sense. And I think as technology keeps advancing, a lot of people are thinking about what the future of automation may look like. So, Vinod, how do you see automation evolving over the next, let's say, even just 5 to 10 years in the anti-doping laboratories?
Well, there will certainly be more of it. The advantages of automation are undeniable, and it's only logical that it'll become more and more pervasive. It wouldn't surprise me to see small-scale automation in order to facilitate storage and retrieval of samples. These systems already exist in large clinical labs. It's just that the work that we do is on a much smaller scale. But if a market exists for it, I'm sure somebody's going to come up with a system that we can use. Or you could have giant sensitivity gains in instrumentation, which means that a lot of compounds could be analyzed without much initial workup at all, which opens the door to one-piece flow and a move away from batch processing. And that almost means a conveyor belt type of workflow from start to finish. Another area that's inevitable, I think, is the use of machine learning to review the data. We generate a lot of data. It takes a lot of time to go through it, but it is really feature-rich and therefore amenable to an AI-based analysis to pull out the positives from the noise of the negatives. The positives are very few. Anti-doping systems generally do have a good deterrence effect. So we're looking at a lot of negative data to find the 2% or so of positives of the samples, and an AI-based approach could help in that initial screening process anyway. Many labs, including ours, are actively working on this in the anti-doping space, and one of us is likely to produce a working model in the next few years.
Very interesting. And I guess from your perspective, are there some tasks that you think will likely either never be automated or certainly not be automated anytime soon that you can think of to give us some examples?
Assays that require large amounts or variable amounts of starting material are hard to automate. And also where you need every last amount of the extraction process are hard to automate because there's always a little bit of waste involved in the automation process. And with respect to review of data, I think I would be very uncomfortable in releasing a data packet that hasn't been viewed and evaluated by human eyes just for the obvious errors that you only know about with experience. So those things I think will still require some human intervention.
That makes sense. And I know in terms of the research side of the work that you're doing there at SMRTL, last fall, you and your colleagues published a new research paper examining minimum reporting limits to determine in-competition use of stimulants. So, Vinod, I'd love to talk about this research that you've been doing. Can you share some of the background on why this study was so important to do?
So in the WADA world, for clients that are WADA signatories, stimulants are only prohibited in competition and not out of competition. So we've had some cases at our laboratory where in-competition collected samples have tested positive for stimulants above the cutoff to report positivity. And the finding has never been challenged. It's clear as day, but it's often difficult or in some cases impossible to conclusively determine whether the substance was actually used in competition or before the competition period started. And there was a lack of literature available in order to conclusively determine this from a urinary level. And we therefore thought it would be worth doing a little bit of a controlled administration study with commonly used stimulants to determine whether the urinary thresholds or cutoffs were successful in establishing in or out of competition use. Another aspect of the study was that there's a lot of momentum in our field to move towards dried blood spots for analysis of these or detection of these in competition substances. And the thinking there is that they're a direct reflection of what's circulating and therefore physiologically available for performance enhancement. So as part of the study, we also looked at a few blood matrices.
I see. And perhaps we could go into some detail then about how the study was conducted, perhaps what commonly used stimulants you looked at.
So we used prescription stimulants that are commonly abused: amphetamine, methylphenidate, and modafinil. I know cocaine does feature in the findings quite a bit, but it would have been a real nuisance to get IRB approval to dose our subjects on cocaine, so we decided not to pursue that one in particular.
Right.
But for the other three, they're available by prescription. We picked relatively low doses, and we tried to have between 4 and 6 subjects in the cohort for each drug, and we tried to include both males and females in each cohort. We used, as I said before, lower prescription strengths. We dosed the subjects once a day for 5 days. And then during this period, we collected samples. And at the cessation of the 5-day dosage, we collected samples every 12 hours for 48 hours. So 2 days. We had 4 collections in the washout period. And with respect to the samples collected, we collected urine, which is the primary matrix for determining these substances now. We also collected dried blood spots, serum, as well as oral fluid, but the oral fluid ended up being not particularly useful for the study.
I see. And I think, Vinod, as you described it, you make it sound easy, just sort of one step after another. But what were some of the challenges in this study? This is a relatively involved protocol.
Generally speaking, the study went very smoothly. We had good volunteers. They were very cooperative. One of the things that might have been challenging was because we drew serum every 12 hours during the washout period. And if we had used a phlebotomist and decided to do venous draws, this would have been problematic because it would have meant people showing up at the lab at 9 o'clock at night and things like this. But we chose to use the Tasso Plus device, which collects capillary serum, and the subjects are able to self-collect at home. So that really simplified the sample collection process.
That makes sense. And perhaps we can talk next about the results. So, Vinod, can you share with us what were the results of this study? And perhaps thinking about those results, what are the implications for the anti-doping field?
When looking at urinary concentrations, at the end of 48 hours, every subject, irrespective of the drug administered, produced a urinary concentration that was above the cutoff for reporting a positive. So if this were an in-competition test, they would have all tested positive by the urinary concentration. And this is relevant because for WADA signatories in that world, the in-competition period begins at 12 a.m. on the day of the event, which means less than 48 hours might have elapsed between the start of the in-competition period and when a sample was collected for testing, which further means that somebody could have used a stimulant outside of the in-competition window during the out-of-competition phase and still tested positive during an in-competition test. So I think we've established now that the urinary cutoffs are not sufficient to distinguish between in and out of competition use. And if we moved on then to the blood matrices, even with DBS, dry blood spots, or with serum, in all the subjects, the drugs were still detectable after 48 hours. Now, you can look at this in two ways. One way of looking at this is to say, oh, well, they still had circulating levels of these drugs, and therefore the urinary concentrations reflect that. And that's possible. But an alternate explanation or an alternate way of looking at things is that, well, we don't know what the physiologically relevant levels of these things in blood is. There's no way to say that just because there was a trace amount circulating in system, the person was deriving any kind of performance enhancement from it. Because these drugs are typically used for ADHD treatment, and there's a wide range of concentrations at which they're effective, and it's very person dependent, it's likely that the level at which a person derives any performance enhancement is also likely to vary from person to person. So really, the only thing we've established with the study is that urinary concentrations are not good to distinguish in and out of competition use. But with the rest of the matrices, we might have raised more questions than we've answered.
That makes sense. And I think that's often the case in science, that you end up with more questions than answers. But what are some of the next steps for this line of research to try to tease out these different variables?
If we want to use some sort of blood matrix in order to determine use of these stimulants, we will need a much larger study, more participants, different doses, in order to come up with a reasonable concentration at which we can say that, well, they're no longer receiving any benefit from this drug, and therefore this is the concentration above which you should report a positive. But that's not a really clear-cut answer, because when we looked at serum and dried blood spots in the previous study, we saw that there was a definite difference between the two. The concentrations in dried blood spots were higher than they were in serum. And they're both blood matrices, so which of these are we going to target for establishing a threshold? That's yet to be answered. If it were up to me, I would probably do away with dried blood spots altogether and just focus on serum. The advantages of dried blood spots are that they can be collected without a phlebotomist and that they can be shipped at room temperature and the drugs are stable in dried blood spots. But we've already shown here that in serum, there are devices now where you can do collection without a phlebotomist. And there's increasing evidence that a lot of the targets we're looking at are stable in serum at room temperature also. The advantage with serum is that you get a much larger volume to work with. So you have the ability to do multiple tests on the same collection. And you might also have enough to do an initial screening and a confirmation after. So if we are serious about using blood, we need a much larger study to determine concentrations or thresholds. On the other hand, you could do away with this altogether and just have a single list that's prohibited at all times, and you wouldn't have to worry about the subtle differences between in and out of competition usage. Some of the professional sports in the United States, like NFL or the MLB, have a single list. It's similar to the WADA list, but they don't differentiate in and out of competition, so stimulants are prohibited at all times, which makes the results management a lot simpler.
Definitely. And it sounds like there's potential that even just making that window for in competition maybe a little bit bigger might help clear things up as well, rather than just starting at 12:01 the day of competition.
Right.
Very interesting. Well, Vinod, thank you so much for talking through this study and sharing some of the exciting results with us. And perhaps to tie it back to our conversation of automation, I know you mentioned automation is not traditionally used in a lot of your research studies. Did you use automation in this study?
We did not. The number of subjects was small, and the number of samples generated could fit in 2 or 3 96-well plates. The extraction process was very simple. For a lot of matrices, it was just a simple dilute and inject. So no large-scale automation was used unless you discount the use of multi-channel pipettes or repeater pipettes in order to achieve the dilutions required.
That makes sense. And we talked about this particular study. Is there any other research project you're working on at the moment or have done recently that you'd like to highlight today?
We're always trying to do more research. A lot of it is driven by the needs of the field. So if there's a new drug on the market where we don't really know what the metabolites are or what the detection windows are, we'll try and do a controlled administration for those. Or we might do washout studies in order to determine whether one metabolite versus another can help you distinguish the route of administration or the time period of use. So we have a few research projects in progress, but we don't have results to share about any of them just yet.
Well, listeners will have to keep an eye out for more exciting research coming out of the SMRTL laboratory. And throughout our conversation, we've talked about the work that you do there, but I think the work is often not done alone in anti-doping. So, Vinod, can you share with us how you collaborate with some of the other stakeholders in the field, whether it's funding organizations like the PCC or others?
Yes, I assume you mean in terms other than hitting them up for funding.
That's included.
Some of the stakeholders in the field also happen to be clients of ours. It's a small universe that way, and some jurisdictional overlap is inevitable. So if some of them are our clients, then we do the best we can to provide the best testing program that we're able to do. Occasionally, they will have need for expedited turnaround, and we'll do our best to accomplish that. Sometimes there are questions about results management, or they need to explore alternate ways in which the athlete could have been exposed to a certain drug. And that involves testing of a lot of supplements or doing even small one-off administration studies to try and clear that picture up. So we're happy to collaborate with our stakeholders in that way. With respect to funding organizations, we try and submit research proposals that are relevant, of general interest to the field, but also aligned with the funding organization's mission and interests and priorities for that particular year. And if we do end up getting funded for a particular project, we're fairly conscientious about taking it through to completion and publishing the results in journals where people can access them and where that data can be useful to the community as a whole. And occasionally we'll also present some of this data at the PCC conference, for example.
Well, that was the perfect segue, Vinod, because you did present at the recent 2025 PCC conference. So can you tell us a little bit about what your experience was like, both attending the meeting and then also sharing your work?
It was a great experience overall. For me in particular, I've been in the field for a little while and know most of the big players, but there are still a few people that have corresponded but by email, but I've never met them in person. So it was nice to put a face to that name and have a few discussions and make those connections. The presentation that we did at the conference was also fun to do. I got to sit on a panel with Ellie Aarons and Mario Tevis, both of whom are colleagues and friends in the field I've known for a long time. So it was really fun. We also got to answer a few questions and exchange and bounce some ideas off of each other. Generally, that was a rewarding experience. But I would like to point out that a lot of the scientific meetings that I go to are all about science, which I do like. But the PCC meeting, this one in particular, also provided the opportunity to listen to some other people. We got to hear from the NLB investigations team. We got to learn about the pitfalls of online pharmacies and the work that was being done in order to uncover black market websites and the efforts being made to shut them down. All of that was interesting. But I would like to also point out, and this was important, we got to hear from 2 previous athletes, which is the community that this work is meant to serve. And as a lab, we don't always get that opportunity. So I really appreciated that. All in all, it was a well-rounded meeting and I enjoyed my time there.
Well, we were so excited that you could attend the meeting and your presentation was wonderful. That panel discussion that you, Ellie, and Mario did was remarkable. So we appreciate you being part of the PCC community. And I think oftentimes there are limitations. We sort of highlighted earlier, funding might be one of them, or even just time, the feasibility of the projects that you'd like to do. So if we took away the barriers that typically hold you back when you're thinking about research, Vinod, what is one problem in the anti-doping field that you would most want to solve?
This may not be a particularly novel answer, but I think that the matter is serious enough that we should address it. I would really like to be able to tell the difference between accidental, inadvertent exposure to a drug and then the tail end of an actual doping regimen. Often the concentrations are small and our instruments are getting so sensitive that we're able to detect at levels that we've never seen before. But that also means that all the other explanations for how the drug might have gotten in there become more and more plausible. The accidental exposure scenario becomes more and more plausible. And the point of anti-doping, in my opinion, is to provide a degree of protection to the clean athlete and incentivize clean sport as opposed to busting people for positives. I mean, the two are related, of course, and you can't have the former without the latter. But if the focus becomes more on this strict liability, zero tolerance, harsh enforcement, then you do run the risk of some of these accidental exposures being also prosecuted, and some innocent athletes are going to get caught up in the churn of the proceedings, and that will probably end up alienating the very community that we're meant to serve. So it would be nice in order to be able to tell the difference between the two and protect the clean athlete in that way and not punish an accidental innocent exposure. I am aware that analytically this may be impossible to do. However, I would like the establishment of some common sense thresholds or cutoffs, at which point it becomes increasingly improbable that this is a trace accidental exposure. Doing that will require a very large administration study using a lot of people and a number of different doses in order to come up with a coherent cutoff. And that's going to be really expensive. But in this world, we have every resource we need. So I think we could do that.
Oh, I like it. Well, Vinod, this sounds like a fantastic dream project. If there's any listeners out there who have the funding for it.
Yeah, please get in touch.
And we appreciate you sharing this project with us today and sharing some of your experiences and your expertise. But if our listeners are intrigued and they would like to learn more about you and what you do, Vinod, what is the best way for them to get more information or to get in touch?
Some of the work that we do is contractually confidential with our clients. So that's not really always available to people, but we make an attempt to publish things that may be of interest to the anti-doping community, but also sometimes to the wider public in general. They can go to Google Scholar or PubMed, where our publications are available. And if somebody is really interested in getting the full PDF of one of our publications without paying for it, they could also get in touch with us. We're happy to send out published material to genuinely interested people.
Fantastic. Well, we appreciate that. Thank you for sharing these resources with me and our listeners. Vinod, it's been such a pleasure to have you on the show today. Thank you so much for your time.
Yeah, thanks very much for having me. I appreciate the opportunity to reach out to your listeners, and I'll be happy to do it again if you need it.
Well, we will keep you in mind for future episodes. And listeners, it's been wonderful to have you here with us as well for this episode. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.